Hello everyone. In the last video, we have discussed about the first type of antigen-antibody interaction, which is precipitation reaction. It is the type of a reaction in which soluble non-particulate antigen combine with soluble antibody to produce visible precipitation complex. Okay, this type of reaction only occur in presence of electrolyte like NaCl and optimum temperature and pH. Okay, so now in this video, I'm going to talk about different type of test which is based upon the precipitation mechanism. Okay, so precipitation is the type of immune reaction because it contains antigen and antibody, and in case of precipitation, soluble non-particulate antigen combined with its antibody. Okay, and after combination of antigen and antibody, a visible precipitate is formed. Okay, now the precipitation reaction can be occur in liquid as well as in gel medium. Okay, so first we will going to discuss precipitation reaction which occur in liquid medium. The well-known example of this type of precipitation reaction is precipitin ring test. In this type of reaction, generally the antigen antibody complex produced in liquid medium. For example, suppose this is the test tube which contain buffer, and in this test tube we have added antibodies at the bottom and their complementary antigen at the top. So these are the antigen and the bottom layer is of antibody, and when this antigen and antibody diffuse towards one another, they react at the middle portion. Okay, antibody get bind with antigen, and due to this binding, zone of equivalence reach in the middle portion, and in this portion we can see precipitate, okay, or precipitation reaction. Okay, so this portion shows actual precipitation complex or lattice formation. Okay, in the last video I have discussed that for any type of a precipitation reaction, the concentration of antigen and antibody must be equal. The reaction cannot occur when the antibody or antigen is in excess. It is called as prozone or pre-zone. Okay, so the portion at the top of this tube contain antigen excess where the reaction cannot occur, and at the bottom of the tube is the portion of antibody excess. So precipitation reaction only occur at the middle portion of the tube where antigen and antibody concentration is nearly equal. Okay, and this type of reaction which occur in the liquid medium is called as liquid precipitation reaction, and the example is precipitin ring test. Why we call it as a precipitin ring test? Because the precipitin or precipitation complex is produced is in the form of ring at the center of reaction or at the center of test tube. So it is called as precipitin ring test. The next well-known type of precipitation reaction is occur in, instead of liquid, it occur in gel medium. Okay, so any type of a precipitation reaction when occur in the gel medium, it is called as diffusion. So remember this thing, when precipitation reaction occur in the liquid medium, the reaction can take place similar to that of the natural precipitation reaction, but when it occurs in the gel medium, it is called as diffusion, because in this case either antigen or antibody get diffused towards one another. Okay, and what is meant by diffusion? We know that the standard definition of the diffusion is movement of the molecule from higher to lower concentration. So same thing happen when precipitation occur in the gel medium. And the example of this type of reaction is number one, single radial immunodiffusion. Okay, so this single radial immunodiffusion is also called as SRID, or sometimes it is called as radial immunodiffusion, that is RID. Another name of this type of reaction is Mancini test. Okay, so all are the same, single radial immunodiffusion or SRID or RID or Mancini test. And the next type of the gel medium precipitation is double immunodiffusion. Okay, and the third type is immunoelectrophoresis. So in this video, I am going to mainly talk about the first type of the gel medium precipitation, that is single radial immunodiffusion. Okay, name itself suggests it is the radial diffusion process. What is meant by radial? And the process of diffusion in which either antigen or antibody diffuse radially in the gel. They diffuse in circular pattern in the gel, so it is called as radial. And the reaction contain antigen and antibody, so it is called as immuno. And it is occur in the gel, so it is diffusion. So radial immunodiffusion. And in this process or in this reaction, antibody remain stationary in the gel while antigen is moving, antigen is mobile, antigen move towards antibody. So only antigen move, antibody remains stationary, so it is called as single radial immunodiffusion. If in the diffusion process both antigen and antibody move towards one another, in that case we can call it as a double immunodiffusion. But in this case, or in the first type, only antigen will going to move towards antibody in the gel, so we called it as a single radial immunodiffusion. Okay, so now in this video, first I am going to talk about principle of the technique single radial immunodiffusion. Okay, in this process antibodies are mixed and remain stationary in gel. Okay, then antigens are moving or antigens are mobile, so we call it as a single radial immunodiffusion. And the process generally carry out in buffer, also called as assay buffer, because this buffer can maintain the pH of the gel, which is very important for any kind of antigen antibody reaction. Okay, now in case of single radial immunodiffusion, antigen diffuse radially in gel, and due to diffusion of the antigen or circular precipitation ring occur in the gel. The diameter of the precipitation ring is directly proportional, or it is directly depend upon antigen concentration. Okay, so by using single radial immunodiffusion, you can easily find out the concentration of antigen in the sample. Okay, so it is a quantitative test. One can use this single radial immunodiffusion technique to determine the either antigen or antibody concentration, as well as the technique can also be used for finding out the purity of the antigen present in the sample. Okay, so all these are the important points that one should remember at the time of studying single radial immunodiffusion. If we want to perform this test in the lab, in that case we need some requirements. Okay, so I'm going to discuss now various requirements that one should need for performing single radial immunodiffusion test in the lab. So first thing that you need is gel. Okay, for making gel, either you need agar powder or agarose or polyacrylamide gel. Polyacrylamide. Okay, so all these are the powder form chemical which is generally used for preparing gels. Then the next thing you will need is standard antigen. Okay, so for preparing standard graph during the practical, we need standard antigen. Generally, 4 standard antigens are used, like standard antigen A, then standard antigen B, C, and D. Okay, and similarly, you will going to need test antigen. Third requirement is test antigen. What is the difference between standard antigen and test antigen? These are the antigens whose concentration is known before performing the assay. So we should know the concentration of these standard antigens. Okay, and the test antigen is the type of antigen whose concentration is unknown. Okay, so before performing the experiment. For example, suppose this standard antigen have concentration 2 mg, 2 milligram per ml. Similarly, the B have 4 milligram per ml concentration. C antigen have 8 milligram per ml, while the D antigen have 16 milligram per ml concentration. Now the concentration of test antigen is unknown, and we want to find out the concentration of this antigen by using the single radial immunodiffusion technique. The next thing that you need is buffer, assay buffer. Because antigen and antibody both are proteins, and proteins require very specific pH for their action. If pH fluctuates during the experiment, the chances of denaturation of the protein increase. So we need assay buffer to maintain the pH constant during the procedure. The next thing that we need is antibodies. Okay, so we need antibodies specific for these antigens. So all these are the important things that you need before performing the practical. Another thing which include Petri plate, then gel puncture for making well in the gel, and all other glasswares. Okay, now I'm going to discuss about actual procedure of this practical. The first step is preparing 1% gel. You have to prepare 1% gel either by using agar, agarose or polyacrylamide powder. And for preparing 1% gel, generally we dissolve 1 gram of this powder. Like for example, agarose. In that case, I am going to dissolve 1 gram of the powder in 100 ml buffer. Okay, so I'm going to weigh 1% agarose powder and I'm going to dissolve it into 100 ml assay buffer. Now, initially the buffer will going to look blur, okay, then or turbid. So we have to boil this mixture so that the agarose get dissolved in the buffer. So the next step is boiling to dissolve agarose in the buffer. After dissolving the agarose, crystal clear solution will be produced, and then the next step is addition of the antibody, addition of the antibodies in the molten boiled agarose. Okay, remember that after boiling, the temperature of the agarose solution will be very high, and at that temperature, if you add antibody directly, the chances of denaturation of the antibody will be increased. So first, you have to cool this boiled agarose to up to 55 to 60 degrees Celsius. And after cooling the boiled agarose or molten agarose to 55 to 60 degrees Celsius, then you have to add antibody so that antibody remain intact in the boiled agarose. Okay, so after cooling you have to add the antibody and then mix the molten agarose solution thoroughly so that antibody get properly distributed throughout the gel. Then the next step is pouring. You have to pour this agar or agarose solution containing antibody into either on the glass slide or in the Petri plate. For example, suppose this is the Petri plate, okay, glass Petri plate, and now in this Petri plate you are going to pour the agarose solution, okay, agarose gel containing antibodies. Now after pouring into the Petri plate, the next step is allow gel to solidify in the Petri plate for 30 minutes. Okay, so allow gel to set for 30 minutes. During this time, the gel gets solidified in the Petri plate. Now remember that this plate contains agarose gel containing antibodies. Okay, and these antibodies are distributed uniformly throughout the gel. If you determine the concentration of antibody at this point or at this point or at the center, the concentration will going to remain same. Now the next step of the procedure is puncturing or making the well or hole in the gel with the help of gel puncture. With the help of gel puncture, I am going to make well or hole in the Petri plate. Just use a small amount of suction pressure to remove the gel from the specific point so that a hole or well will be produced in the agarose gel. Okay, now in this well I am going to add my antigen, both standard as well as test antigen. Okay, so the next step is addition of antigen. Okay, so I'm going to draw the same plate again. For example, this is the Petri plate which contains agarose gel with antibody, and now in this Petri plate I have make the wells with the help of well puncture. For example, this is well number 1, well number 2, well number 3 and well number 4. I have also made another well at the center. Okay, and now in this well I have added different antigens. For example, I have added standard antigen A, B, C, D in these wells. In the 4 wells which is outside contain antigen A, B, C and D. Okay, so 4 standard antigen I have added in these 4 wells, while the remaining 1 test antigen I have added at the center. So this center well contain test antigen. Okay, and at the time of addition of this antigen with the help of micropipette, I should keep this plate horizontally on the flat surface. Now after addition of the antigen, the next step is incubation of this plate in moist chamber for 24 to 48 hours. Okay, 24 to 48 hours. So then I have to keep this plate in upright position in the incubator at 37 degrees Celsius for 24 to 48 hours. But the care should be taken that at the time of incubation, the gel may get dried, and to avoid the drying of the gel, we should use moist chamber. Okay, if in the lab moist chamber is not available, in that case you can simply put this plate or simply keep this plate in a chamber like in plastic box, and the box should contain cotton balls soaked in glycerol. Okay, you have to just make the cotton ball, soak them in the glycerol and keep this ball in this chamber. This glycerol cotton ball will going to maintain the moisture inside the plastic box which contain this petri plate. Okay, moisture is very important for proper diffusion of the antigen in the gel, so that once the antigen get diffused, the reaction will going to appear properly. Okay, so the next, after incubation, what happened during the incubation? For example, now this A, B, C and D are the standard antigen whose concentration is known, like 2, 4, 8 and 16, 2, 4, 8, 16 mg per ml. Okay, so now in each well the antigen is present which start diffusing radially into the gel. Okay, so antigen start diffusing radially into the gel. The diffusion occur like this in all direction. Initially, close to the well, antigen molecules are present in excess, and we know that for any type of a precipitation reaction, antigen and antibody should be present at an equal amount. Okay, so antibodies are equal or antibodies are the same throughout the gel, but near the well, near the each well, antigen start diffusing and their concentration is too high, so that precipitation reaction do not occur. Okay, during the incubation, the diffusion of the antigen occur away from the gel, and once they reach to the sufficient distance, the concentration of the antigen goes on decreasing. Okay, for example, this is the well which contain antigen, and due to the diffusion of the antigen through the gel, their concentration goes on decreasing, and once their concentration reach or they become equal to the concentration of antibody present in the gel, they stop migrating. Antigen antibody complex produce, zone of equivalence occur, due to which lattice is produced, the precipitation complex is produced, and due to which a visible reaction occur. For example, due to the diffusion of the well, antigen in the well C, now the line is occur somewhere here. Precipitation is occur or observed as a white line, white precipitation line around the well. Okay. Now remember that the diameter of this precipitation ring, which is also called as zone of precipitation, is totally depend upon concentration of antigen present in the well. More is the antigen in the well, larger will be the diameter of the precipitation ring, and vice versa. Okay, so now this A antigen have concentration 2 mg per ml. So this A antigen will going to give quite smaller zone of precipitation. B antigen have concentration 4 mg per ml. So this B antigen will going to diffuse and going to produce somewhat larger zone of precipitation. C antigen have antigen concentration 8. So this will going to produce more bigger precipitation zone, and finally D antigen have concentration of 60 mg per ml. So D antigen will going to produce largest zone. So here you can see the diameter of precipitation ring or precipitation zone goes on increasing when we move from antigen A to antigen D, and this is happened because of the concentration of antigen. Okay, so remember that the area of the precipitation ring or diameter is totally depend upon concentration of antigen. And now with the help of this 4 standard antigen precipitation ring, I can make standard graph. Okay, so for plotting standard graph, I have to just take a graph paper, okay, and on the x-axis I'm going to take antigen concentration, and on the y-axis diameter, okay, diameter in mm. So the y-axis have precipitation zone diameter in millimeter, and while the x-axis have antigen concentration. We have used 4 antigen concentration, 2, 4, 8 and 6. So I am going to make 2, 4, 8 and 16. 2, 4, 8 and 16 mg per ml antigen concentration point on x-axis. And then I just have to make the point, okay. So diameter is like this on the y-axis. So for example, antigen A will going to give say 5 mm zone. So the point will be like this, near to 5 mm. Then the B antigen will going to give another point, C antigen, and finally D antigen. So you will going to get point pattern like this, and then with the help of this you can easily draw standard graph, straight line. Now this graph can be used readily for finding the concentration of test antigen, and for that you have to just measure, for example, the center well contained test antigen, and that have produced precipitation zone like this. So you have to just measure the diameter of the precipitation zone with the help of scale of the test antigen. And for example, if it is giving say 12 mm diameter of the precipitation zone, then you have to just point or find 12. For example, this is the point on the y-axis which have 12 mm. Now you have to just extrapolate the value of this test antigen diameter on the x-axis, and it will going to give you a point on the x-axis which exactly give you antigen concentration for test antigen, okay. So just by measuring the precipitation zone diameter of the test antigen and by extrapolating it on the standard graph, this is called extrapolation. On the standard graph, you can easily get the concentration of test antigen with the help of x-axis, okay. So in this way, by using the procedure, you can find out the concentration of any unknown antigen by using single radial immunodiffusion technique. It is called as single because in this technique only antigen is moving and antibody remains stationary, or antibody is in the gel which is unable to move. So it is called as single radial immunodiffusion technique. The technique have very wide application in hematology because we can easily find out the concentration of any type of antigen from the blood sample or from the serum sample with the help of this technique. Only thing you need is antibody specific for that particular antigen. Thank you.